Really?
Okay? Not okay? So remember we talked about the last thing I did with the simple columnar epithelial cell, and we had the micelles, the biosalt, and then breaking down with lipase, and then we had it moving into by simple diffusion across the plasma membrane, reforming into triglycerides. Think about what happened to it after that and where it would have gone. Okay? It went into the lacteals, not into the capillaries. So that should give you a hint. We follow that through all the way to the liver afterwards. All right? So let's look a little bit. We're not going to go through all the steps of the citric acid cycle. That would be a whole other hour lecture. All right? But I do want to do some highlights. So our physiology, the metabolism quiz is an extra credit quiz online. So there's a lot more material in your lecture notes that you can use for that quiz that I'm not going to cover on here. But what I cover here will be what you would be responsible from that for the exam on Tuesday night. Okay? So when we talk about most of our body source for energy comes from carbohydrates. We can bring lipids and amino acids into these three processes as additional sources, and we'll see that when we look at the hormones. But primarily, this is set up to use carbohydrates, all right? Glucose, and then the glucose is converted to fructose as we start out. So we have these three different steps. This, by the way, for metabolism. Metabolism is a two-step process, well, two-component process. If we're breaking material down, like we're breaking glucose down to its carbons and electrons, that is known as catabolism. I always think of catastrophe. Catabolism is breaking apart. Okay, anabolism is building. If you think of anabolic steroids, that's for like making more muscle and stuff like that. So catabolism is the breaking apart components. So taking glucose and releasing carbon dioxide and heat and capturing the energy. So for catabolism of glucose, or at least any of our simple sugars, because the glucose is soon converted to fructose, we have glycolysis as the first step. All right? And this is considered to be anaerobic. It will take place, if you learn to remember from Bio 430 in muscle, this would last for about 15 to 20 seconds. The creatine phosphate, you would get energy from about 2 seconds, but you need aerobic metabolism to go longer than that. So this is considered to be anaerobic. It takes place even if the cells do not have sufficient oxygen. It also takes place within the cytoplasm of the cell. So you can see up there is a diagrammatic version of the mitochondria. But glycolysis does not take place in the mitochondria, it takes place in the cytoplasm. Right? And it's very limited. It only makes the net gain is 2 molecules of ATP. It actually makes 4, but we have to prime the pump, so to speak, with a couple, so our net gain is only 2. Of the entire process that's up there on the board, there's a total of 38. So you can see how limited anaerobic activities are to provide ATP. Now, this is modified each step. We add phosphates, we break carbon-carbon bonds, we capture some hydrogen atoms. Each step is regulated by an enzyme, okay? And so enzymes, I said, were proteins, or we agreed together they were proteins. So proteins are synthesized via messenger RNA that comes out of the nucleus from genes that code for them. So the messenger RNA then associates with ribosomes. Would that messenger ribosomal cluster attach to a ribosomal ER, or would it just be forming 3 ribosomes within the cytoplasm, what we call little clusters or polyribosomes. 3, because these enzymes do not have a membrane-bound destination. They're being used within the cytoplasm, so we're not sending them out of the cell, we're not sending them to the mitochondria, they're interacting in the cytoplasm itself, all right? So these would be using synthesis, enzyme synthesis would involve polyribosomes or 3 ribosomes. All right, so in this step, we have 6 carbons. They're in a chain form, all right? And by the time we start the step, and I have a double arrow, but it's not every step is reversible, we end up with 2 3-carbon molecules. What do we call the 3-carbon component of our lipid? Triglycerol. The 3-carbon part was the glycerol, okay? So guess where triglycerides can come into if we're trying to use fat for energy instead of glucose. This is where the glycerol part of our triglycerides can be utilized in place of glucose. All right, and then that 3-carbon component. Now, we have some release of energy, enough to make our 2 molecules of ATP. We also release some hydrogen molecules. Well, those hydrogen molecules are carried by a molecule known as a coenzyme. It doesn't enter into the chemical reaction, but it's going to transport this hydrogen to the mitochondria. Okay? Now the next step is called the citric acid cycle. Krebs cycle is an old term. So this takes place in the mitochondria. So guess where these enzymes, how these enzymes are made. They have a membrane-bound destination. So the enzymes for the citric acid cycle and the enzymes for electron transport are going to use ribosomal ER and Golgi to make their enzymes. All right? So this is aerobic. It requires oxygen. It will not occur without oxygen. And it takes place in the mitochondria. And the enzyme synthesis occurs via our ribosomal ER and our Golgi. Okay? Because our mitochondria are enzyme bound. Now, so we bring this 3-carbon molecule towards the mitochondria, into the mitochondria. Along the way, we lose a carbon, the intermediate step. Guess what? That carbon takes with it some of the oxygen that was part of our original glucose or fructose molecule. That's where the carbon dioxide comes from that we breathe out. Okay? It's a release of carbons from our food stuff. Normally our carbohydrates, but if we're using triglycerides in there, or we're using amino acids, it's the carbon of that food that we've eaten that we're releasing as carbon dioxide. Okay? So when we lose a carbon, what actually comes into our mitochondria is a 2-carbon molecule. Within the inner matrix, we combine it with citric acid, which happens to be 4 carbons. And then it goes through a series of reactions, and we end up back up with, actually, misspoke. This is oxaloacetic acid. I said it wrong last night, which is 4 carbons. And when the 2 molecules come together, we have citric acid. The reason it's called a cycle is it's not like a fairy ring of enzymes, but as we go through this, we end up back up with oxaloacetic acid, okay? So we go through a series of chemical reactions giving off different products, and then different, even ATP, GTP is produced. And along the way, we have to do it twice because we have 2 of these coming in. This is where we lose our last carbons, is carbon dioxide. All right? So more carbon dioxide is released. And we're doing this twice. So we have 6 carbons of carbon dioxide released for every glucose molecule that we break down. So what are we left with? Our hydrogens, okay? So that brings us to our electron transport. Yes?
So what are the enzymes?
Where?
What are these enzymes?
Okay, so those are what we're going through. So enzymes, I'll show you some enzymes for electron transport. Enzymes are, the purpose of an enzyme is to increase the rate of a reaction by straining the bond, okay? So in the cytoplasm, we have enzymes that, this is what you don't have to know, that add a phosphate group here and a phosphate group here and break this bond. The enzymes don't, but they strain the relationship of water to break that bond, all right? And then each of these is a separate step regulated by an enzyme that binds to that and does that specific chemical reaction with it. In the mitochondria, we have an enzyme that regulates the binding of the 2-carbon group here with the 4-carbon group here. Then we take off hydrogens or carbons, and we go through a series of different, and you have the diagrams in your lecture notes, but hold on just a second.
So do we need to, where they're synthesized, that's it, the enzyme?
What I want you to know is essentially what I have on the board. Okay, so where the, hold on just a second here. I don't have one. This is what I was cutting out. So here we start off, and you still don't have to know it, but to explain your question, here we start off with the 6 carbons of glucose, okay? We add a phosphate group to each end. That's regulated by an enzyme. Then we take that 6-phosphate group and we break it into 6-carbon group, and we break it in 2. That's another step regulated by an enzyme. And then we remove hydrogens, regulated by an enzyme, okay? So each of those components is regulated by an enzyme. And here's the citric acid cycle. So what I just drew in the board was we have our 2-carbon group coming in, adding to this 4-carbon oxaloacetic acid to form our 6-carbon citric acid. There's another enzyme. Then we modify where the carbons are. We remove some hydrogens, we lose a carbon, another enzyme regulates that step. And then we remove more hydrogens, remove another carbon, another enzyme. So we go through these series of chemical reactions producing some ATP, ending up finally with what we started off again, ready to accept another 2-carbon group. That's why it's called a cycle. Okay.
So we need to know that the enzymes regulate.
So that you need to know that enzymes are present in the middle of the mitochondria for the citric acid cycle, and that those enzymes are produced by the cell using the ribosomal ER formation, because the mitochondria is a membrane destination. Okay? So once we're done with the citric acid cycle, we have all of these coenzymes that have picked up the hydrogen, either in the cytoplasm or in the mitochondria. And that's what we start with the electron transport cycle. Okay? So glycolysis brings our 2-carbon molecule citric acid cycle, gets rid of all the other carbons, and we end up with the hydrogens. Okay? So now we're ready for, so far we still only have two, right now though we have 4 ATPs, but you don't have to know the actual numbers. And that brings us to our electron transport. So electron transport takes place on the inner membrane. And in Bio 430, we talked about how that increases the surface area, plus it localizes it, so that it increases the chance of the substrate interacting with the enzymes, because the enzymes are stuck in the membrane. So they're not wandering over here and over here and over here. They only have to interact with that one structure. So we have electron transport. Well, we don't have any electrons yet. We have hydrogen. How many protons in one hydrogen atom? What's its atomic number? 1. So it has 1 proton and it has 1 electron. So when we separate those two, we end up with a hydrogen ion and an electron. Okay, and that's how we talk about electron transport. So the purpose of this, remember, is to capture energy. The energy was not found in our carbon-carbon bonds. The energy was not found to much extent in the carbon-oxygen. We only made a few ATP molecules. Most of our energy comes from the hydrogen ion, the electron from our hydrogen atom. Okay? So if I go back to this previous image, the hydrogen bound to the NAD is at the highest level. If you imagine a slinky going down the steps, okay, potential energy as it falls down the steps. If we tried to get all of the energy out of that hydrogen here to here, it would be too explosive for the cell. So we go through a series of small steps, releasing a little bit of energy at a time. And that energy is used. Imagine a water tower, all right, and we're going to use a little bit of energy to push water to the top of the tower. And then if we let it all down at once, it's going to spin a turbine. And we're going to use that to create electricity. And essentially, that's what we're doing in the mitochondria. We're releasing a little bit of energy at a time from the electron as it goes from one molecule to the next. So wherever you see these channels here, and that's moving hydrogen ions from inside the cell against their gradient to this outer matrix. There's also hydrogen ions out here already, so it takes some energy to move them. Each molecule, notice the electron is going from one molecule to the next. Each molecule has a higher affinity for that electron, meaning it has a greater attraction for it, okay? But it needs less energy to hold on to it. So, I told the class last night a story. It's not very complimentary to women, so my apologies to our gender. But let's say that there's this woman here that's the hydrogen, the electron of this hydrogen ion. She goes into a bar. She's single, and she meets this guy who's really interesting to talk to. My daughter only picks up guys in bars. All of her boyfriends come from country music bars, and I have no idea why. The last 5. But anyway, I'm not talking about her. None of them have been wealthy. And she goes home with him. They actually fly home in a helicopter. And he's got like 5 Jaguars in the garage and another 6 on the other side of the island. And when she wakes up in the morning, there's 8 2-carat diamonds in the bottom of her goblet of orange juice. I'm like, wow, this is nice. And the guy's fun to be with to boot. So he hands her a bunch of platinum credit cards and says, my helicopter's at your disposal. I'll be back in 3 or 4 weeks. Just go spend whatever money you want. So she does, and she's having fun, but she gets lonely. I mean, 3 weeks is, all by herself. So after about 10 days, she finds herself back at the bar. She took his helicopter to get there. And she meets another guy. He's a lot more interesting. He pays more attention to her. She finds herself attracted to him even more, and she goes home with him. Now, he only has 2 Jaguars. And the next morning, there's only 2 diamonds in her orange juice, and they're only 1 carat. He has as much money as the first guy, but she's so attracted to him, she doesn't even know this, that he's not spending as much money on her. He's socking it away into investments. finds herself back at that bar. It's worked out well so far. And she meets another guy. Goes home. He doesn't have, he only has one car. It's not even a Jaguar, a Ford. And the orange juice has no jewels in it at all. And he gives her one, plain old American Express card. But he's home all the time. And they go horseback riding, then they go surfing, and again, she has no idea that he's got as much money as the first guy. He doesn't have to spend it on her because he's just so much fun to be with. Okay, greater chemical attraction without needing the energy. So that energy that's not needed to hold on to the electron is used to pump hydrogen ions out into the outer matrix. All right? So this just keeps going on. She passes herself from one guy to the next. I told you it wasn't very complimentary. By the end of the year, she finds herself on a shack on the beach. No money whatsoever. She doesn't have any guy to know that the guy actually owns all the islands and is a multi-billionaire. She's just having fun. And she doesn't need all that money because he's just fun to be with and he's there with her. All right? So finally, at the end of this sequence, where the electron is moving to a lower and lower energy level, giving off energy which is used to move the hydrogen ions up here. All right. Then the last molecule to be involved is actually a turbine. We have a protein turbine. It has a channel. And the hydrogen ions go from high concentration to low concentration and spin this molecule known as ATP synthase. And the energy is used to force that third phosphate onto an ADP molecule and make ATP. So little bits of energy released at a time finally come together to create ATP. Okay? So electron transport is our final stage. This is where we make most of the ATP. So this is in the inner matrix for the citric acid cycle. This is in the outer matrix. And at the very end, as the hydrogen ions are coming down through that ATP synthase, they're reunited with the electron that's reached the end of its travels here. So we have a hydrogen ion plus an electron being reunited. All right, a couple of those, and oxygen. Where did our lady end up living? In a shack on the beach next to the ocean, which is made of water. All right, so at our final stage here, we make ATP, we make water, and heat. Because only about 50% of that energy is used on that ATPase, and the rest of that is okay? That's what I want you to know for this process. So what happens at each level, which one is aerobic? Electron transport is aerobic as well. Because neither citric acid cycle nor the electron transport will occur without oxygen at the end of that to be able to accept the hydrogen ion and electrons. Remember our potassium cyanide? It poisoned the cell so that it was not able to accept the electron. So the cell that was poisoned by, any of the cells poisoned by the potassium cyanide can't use oxygen. And that's why when the venous blood gets back to the lungs, it's high in oxygen because the cell was unable to use it. And low in carbon dioxide because this process wasn't completed. Okay. So let's look at some terminology. This is more of a nutrition component, but it comes along with metabolism. 1 gram or 1 mil, this is distilled water, is the same. So may I erase the board? So when you look at the nutrition information on a loaf of bread, and it says a slice of bread typically has about 90 calories. Are we talking about a little c or a big C calories? The big C calorie is the same as a kilocalorie. Okay? So an uppercase C calorie is actually referring to 1,000 calories. So let's put our definition up of a little c calorie first. So this is the amount of energy or heat needed to raise, whoops, get H in there, the temperature of 1 mil of water, and this would be pure water, 1 degree centigrade Celsius. All right? A kilocalorie, a large C, and again, this is what you would read on nutritional information. So could I modify that now to say a kilocalorie would be the amount of energy needed to raise the temperature of 1 mil of water 1,000 degrees? Why not? 1,000 times more than the first one. Water boils at 100. It's never going to get to 1,000. Okay? So instead, a kilocalorie is the amount of energy needed to raise the temperature of 1,000 mils of water 1 degree, or 502 degrees. But we can't raise the temperature over 100. Needed to raise the temperature of 1,000 mils, or a liter, okay? Just 1,000 times the energy, but we can't go to 1,000 degrees. All right. Basal metabolic rate. Now, if you read a lot of nutrition books, they'll tell you that everybody's basal metabolic rate is about 1,200 calories a day. Not true. This is the amount of energy, I'm not going to write it out. This is the amount of energy that you need to do the bare minimum to stay alive. So not to study for your test on Tuesday, all right? Not even to walk or chew food or to blink your eyelids. It's just to be just what your cells need to do to pump and breathe and do their normal daily activity. Nothing else. Not even to think about what they're doing, okay? For some people, that might be 1,200. For other people, it's 800. So there's people that lose weight on what would be the basal metabolic rate for one person, or gain weight on what would be the basal metabolic rate for another, okay? It's usually measured by oxygen consumption, just for the bare minimum. And then if you are cleaning house or running a mile or whatever, that gets added on top of that for your daily calorie requirements. Which brings us to diet. So mal means bad, like malaise, feeling sick. So nutrition and nourishment. Nourishment has to do with calorie intake. Nutrition has to do with the balance of the food that you eat, all right? So if your body basal metabolic rate plus whatever you're doing comes to 2,000 calories, and you take in 2,000 calories, you do not have malnourishment, okay? If you need 2,000 calories and you only take in 1,000, then you're malnourished. Or if you need 2,000 calories and you're taking in 4,000, you're still malnourished, okay? Just on the opposite end of things. All right, what about nutrition? If you're taking in those 2,000 calories, but 98% of them are coming from lipid and not carbs and proteins, that's malnutrition, all right? Or if you're not getting an adequate amount of vitamins. So that's a balance of the content of what you eat, all right? So number of calories for nourishment, balance of what you eat for nutrition. Of course, you can have both. You can be eating too much food and having an imbalanced diet as well, okay? But if you take all your vitamins by pills and you eat protein bars and you're getting the minimum daily requirement of everything, but you're not getting enough calories, you're still going to be malnourished. Okay? All right? That brings us to our last question before we look at hormones. I guess I should put A, B, C, D beside those. I'll just say it. So A would be the first one, B the second. Grandma's A, the pilot, the granddaughter is B, the baby is C, and her pilot's twin sister is D. So if the plane crashes, and they're all dead in the plane crash, which of them would have the best survival chances, given limited food, limited water, and no food?
Because they need more. The 9-year-old is not going to survive because
Sorry.
A black something or another? Yes.
No.
It looks like a pine needle, but there's no pine needle.
Oh.
Did I have an expression on my face?
Yes,
definitely.
Sure did.
All right, so if you chose A,
I think it would be B.
I got two of them up there. She's not going to work out very well because her hypothalamus is no longer working to help control her body temperature or her fluid regulation. For the 30-year-old granddaughter, she has a lot of protein, all right? Protein provides 4 calories per gram, but there's a drawback. She needs a lot of water to help flush out the urea and the ammonia that come from breaking down those proteins, okay? The baby has the same problem with the hypothalamus as the grandma, only his is immature. So the sister, lipids provide double, more than double the amount of energy. All right, so you need a reason. We're done with Thanksgiving, but if you haven't recovered from that, we've got Christmas coming along. If you find yourself flying over the desert, feel free to imbibe in all the eggnog you need. Or just so hard. All right, so our last component then, before we move to our wet lab with digestive, is looking at, we're looking at insulin, glucagon, growth hormone, cortisol, and thyroid hormone. Okay?
Are you going straight to
So before you take notes, let me talk. I have everything summed up for a table at the end. Okay? So we already talked about insulin being released in response to elevated levels of glucose. All right? And what insulin does is it adds, remember the aquaporins we talked about in the kidney? Well, there are glucose transporters, and insulin functions as a hormone to increase the glucose transporters that are inserted into many, although not all of our cells. And so there's going to be a decrease, obviously, in the glucose because it's moving into the cells. So we're going to see a decrease in the amount of glucose. All right. There's also going to be a decrease in lipids. In fact, this is the only hormone that results in the decrease of all three: lipids and glucose and fatty acids. So let me pause here because there's some few words that I want to put on the board. And unfortunately, they all start with a G. Remember the first G we had was glycolysis, okay? Glycolysis is just the catabolism of carbohydrates. So that was the glucose getting broken down, all right? However, as you'll see when we look at these hormones, the body can use other components. So amino acids acid cycle. Remember the fatty acids I described as long chains of carbohydrates? So when we start to break those down, we have these two carbon groups. Remember where we saw two carbons in our brief overview of metabolism? As the glyceraldehyde was coming to the mitochondria, we clipped off a carbon, so these fatty acids could come into the citric acid cycle there. So when we use other foodstuffs for energy, that process is known as gluconeogenesis. Okay? So alternate energy sources for glucose. Lipids or amino acids or whatever. Now insulin causes glucose to be taken up by cells, and the liver also takes up glucose, and what does it form with the glucose in the liver? All those little black dots I showed you, glycogen. So when we make glycogen, that term is glycogenesis. Alright? That would be an absorptive state process where we take excess glucose and store it as glycogen. So this process is glucose to glycogen. Now under the influence of glucagon, we take glycogen and we turn it back into glucose. So instead of glycogenesis, that process is known as glycogenolysis. Long word, it just means we're splitting up glycogen. Okay? So this means where genesis is beginning, we're making glycogen. Glycogenolysis means we're splitting it up, like hydrolysis. So insulin is going to have glucose move out of the blood. It's going to take fatty acids out of the blood as well, all right, to make lipids. And it's going to take amino acids out of the blood to make proteins. So as I said, this is the only hormone that decreases the level of all three amino acids, fatty acids, and glucose.
Does glycogenolysis happen in the liver?
Yes. So the opposing hormone to insulin is glucagon. Have you ever heard of hypoglycemia? Someone that is hypoglycemic that's been making not enough glucagon. So glucagon releases glucose into the bloodstream. Doesn't really do anything on amino acids, but it's going to it's going to cause glycogenolysis. So we're going to see more glucose in the blood. And it's going to cause fatty acids to be broken down, so we see more fatty acids. So both of these are available for energy. Okay? Like I said, I have a table for you in the last one. So insulin and glucagon have opposing effects, both made by the pancreas. I want you to know what organ makes these hormones as well. All right, then we're back to the adrenal gland. The adrenal gland make ADH or aldosterone? Adrenal gland makes aldosterone. Okay. On the lecture exam, quite a few of you, when I asked for a hormone made by the kidney, probably at least half the class took ADH or aldosterone.
Give us the answer, right? Give us some.
Sure, I was looking for one of two, EPO, erythropoietin, and the other one was vitamin D or calcitriol, which is its final conformant. Renin is an enzyme. That's another one. So in the adrenal gland, all right, see in the middle of this where we have all these lipid droplets that you learn to recognize as being in the adrenal gland. This is where cortisol is produced. We're not using the aldosterone here. Cortisol is a stress hormone. All right, it typically blocks the immune system And if there is a tumor, we talked about Cushing's disease a little bit at the beginning with the pituitary gland and ACTH and all of that. So if there's a tumor with the adrenal gland producing too much cortisol, the individual tends to have very skinny arms and legs. That's because cortisol spares glucose and increases the use of proteins for fuel. It's not a good weight loss drug in that sense because you're going to get weak from the loss of protein. It does break down fats, all right? So we end up increasing the level of amino acids because it's breaking down the protein. That's why your arms and legs get skinny and weak. You do lose weight, except in the around the face and the bottom of the neck, because you are breaking down fat. So those fatty acids are going to go up. So both amino acids and fatty acids go up, and glucose goes up because it's not being used. All right, so it's being spared. So we use amino acids are broken down and fats are broken down to be used, but that temporarily increases their level in the blood, and then glucose is up. So cortisol is going to cause an increase in everything, whereas insulin caused a decrease in everything. Okay? I don't want you just memorizing that table, kind of explaining what's going on here. So our next one is growth hormone. This at first glance seems to be the ideal weight loss. So anabolic steroids are used by weightlifters and bodybuilders, and you can hear them being sold on the radio on Sunday mornings for precursors to testosterone and growth hormone to help you lose weight and so on. Because it takes out amino acids and you build, that's the opposite of cortisol. Your amino acid levels go down because you're building up muscle. Fatty acids go up, using that for fuel instead of glucose. So you're making muscle, so your amino acids are removed from the blood. You're breaking down fat, so your fatty acids go up. You're using that for energy instead of glucose, so those levels remain high. However, people who use anabolic steroids for an extended length of time are at risk for diabetes because of that constant exposure to the pancreas of high levels of glucose. Okay? So the pancreas ends up running out of not being able to make additional insulin, or you lose your sensitivity at the receptor site because insulin is constantly being dumped out of the pancreas in response to these continued high levels of glucose. All right? So amino acids are low, the other two are high. Again, sparing, not using the glucose so it stays high. Using fats, the fatty acids are broken down so they become high because we're dumping them into the into the bloodstream. But we're making proteins so that becomes low. Alright, and then finally thyroid hormone. Thyroid hormone increases cellular respiration of our cells. It increases the mitochondria making ATP. So obviously it's going to need more fuel for that. Alright? So this is backwards from what I said last night. It lowers blood levels of glucose and breaks down fats as additional sources for energy. Okay. So here's your table. So insulin decreases all the way across the board. Alright. Cortisol increases all the way across the board. So this is our last lecture slide. A summary of those digestive hormones. Okay. Now your lecture notes do talk about some diseases, but I won't have any questions on the lecture exam about diseases caused by too much myxedema or goiters or that type of thing. Okay. Okay, can't be my last word of lecture, so
The thyroid.
yes, we make more ATP. Alrighty, so how about we be done? So take a few minutes, 5-minute break, and then come back and we'll start our wet lab. If you need the 3 goggles, they are under the hood here. Okay? Yes. So on Tuesday, I'll probably post it tomorrow. There'll be 3 parts. The fat digestive will basically what we talked about today, what we do in lab today. And then the protein and carbohydrate lab part will be on physio. Okay?
And then the lab assignments, and then optional lab.
All righty?
There's a lab. Yeah.
So,
She hasn't posted it yet.
when you come back from break, if you're going break, each lab team